Thank you for the kind words. You know, I was thinking about this talk, and you know, it hit me that the LeukoStrat CDx, I think, embodies our mission, in that Invivoscribe improves lives with precision diagnostics. And what I hope to sort of show is that this assay sort of embodies like our culture and how we deliver precision medicine on a worldwide scale. How I want to do that is, I think, to understand a test, a clinical assay, you have to understand the sort of the why, right? The clinical, the assay, the guidelines, the underlying biology of the cancer or the condition, and then talk about prevalence, right? Prevalence has a big impact on what kind of tests work in a clinical space. So we're going to talk about how AML is unique and in many ways from other cancers and other conditions. And that has implications with assay adoption on a worldwide basis. We're going to talk about the therapeutics, which AML, as you know, has really not had much for many years, with really just conventional chemotherapy is 7+3. But over the past few years, it's been an incredible, exciting space, largely out of the work from many pharmaceutical companies, and also understanding the underlying genomics of AML and other cancers. Then we're going to get into the nuts and bolts. The FDA approved this LeukoStrat CDx assay. Talk a little bit about allelic versus allelic ratio versus signal ratio. And there is a distinction there that's not often talked about in the general public, and also when you read publications. And then finally talk specifically about the Ratify trial and this cross-validation quality control trial that was done as part of Ratify, and talk about our own efforts and working with the FDA and some of the evidence there. So the ELN and the NCA guidelines have recently had new development in binning AML. And putting different patients in different risk categories is critical because it helps determine the intensity of the therapy. So conventionally, they go through like a 7+3 induction chemotherapy. And then historically, if you have a core binding factor AML, basically if you have like this translocation with RUNX1, RUNX1T1, or the translocation version is CBFB, MYH, and biallelic mutated CBP alpha, often those patients don't go on to transplant, and many of those people can be cured. Now, what's interesting in a new development recently was the addition of mutated NPM1 without FLT3 ITD or ITD low. And that's something that is new relatively to the guidelines. And that's what makes, I think, the signal ratio critically important, because potentially oncologists will base their therapy after induction chemotherapy and remission about decisions and potentially about bone marrow based on the signal ratio. But I think what this speaks to is, I think all of us can appreciate that this is maybe an old paradigm, that making decisions based on small binary decisions may not fully encompass what we should be offering patients. And I think some of the most innovative, interesting publications that are coming out are using machine learning or other techniques to decide how to bin people in better risk. And you see that in other diseases like MDS and some other conditions. In AML, there was some interesting work in the UK that they were doing that as well. So, but getting back is, I think it's critically important that you have a signal ratio that people understand. But there's still, even though they made that recommendation, within guidelines, there are a lot of people as part of the NCN network, right? And I don't think everyone reads the same publications the same way. So even within the guidelines, even though it made it up, it's interesting, they say low allelic ratio under 0.5 or 0.5 and above. One group of sort of oncologists say, regardless of the FLT3 allelic ratio, patients should be considered for bone marrow transplant. That they're considered either intermediate or high risk. And even though it says good favorable, everyone goes to transplant. However, there was it seems like another group within the room that said, though recent studies indicate that AML with NPM1 mutation and FLT3 ITD with low allelic ratio may have a more favorable prognosis, and patients should not routinely be assigned to allogeneic transplant. So whenever there's a controversy like that, you gotta basically look at the publications. And what you see is unfortunately, you have a lot of groups sort of develop their own assays, and those assays may or may not deliver the same value of signal ratio. And it becomes critically important when you're dealing with a small, relatively not very prevalent cancer like AML. And so what you see in the literature is, for example, the UK wants to stratify signal ratio according to a certain gradient and base decisions on that. Whereas a Spanish group, Corona, this is a study of about 300 people. And this showed that if you have NPM1 somatic mutation and you have a low FLT3 ITD, or you basically are in a low risk, an allogeneic transplant doesn't offer any benefit. And in fact, you can see, unfortunately, the people that got transplanted, as you could potentially imagine, there was unfortunately a higher risk of death in that first year or two, and then it sort of just balanced out. So you may even be harming people with that type of condition. And so there's a lot of debate after the 2013 paper came out. And Lynch in 2014 had 2,000 patients, and they found an opposite conclusion, that they didn't see that same trend. And Schleck, which we're going to talk about, basically just shows that if you have a FLT3 ITD low, you basically wouldn't benefit from a transplant. So there's a lot of differences here. And some of the issues may be that one group has their own assay, another group has another assay, and they're not completely equivalent. And if you don't have a universal assay, it may be difficult to essentially sort of get through these weeds. But here, this is a German group that basically did this. It's a log-rank statistical method where you first determine how do I separate the two groups, the distributions maximally? And they found out that 0.51 was the allelic ratio that worked best. And that's why many groups sort of jumped on that 0.51 based out of this study. Also, you see clear distribution in survival with overall survival based on the signal ratio. So the signal ratio is really critical. And if you read papers in AML, you'll often be directed to the Schleck paper. And what they see here is really fascinating, right? So low ITD signal ratio, basically if you take people to transplant, what happens? There's basically really no difference, right? And then if they have a high ITD, both overall survival and relapse-free survival, basically there's a marked difference. So it kind of makes sense, right, a little bit, that it's an activating mutation. It happens downstream from the AML biology. And you think if you could just tap that down, that people would survive. And maybe if the AML is not as aggressive, maybe the transplant isn't as needed as much. But there's still a lot of debate, and it's still ongoing. So here's the key is you can have, when there's a lot of questions, and if you're in a cancer like lung cancer, when there's like 200,000 people on average die of lung cancer or diagnosed every year with lung cancer, you can maybe as a single institution do large powered studies. And you can get studies quickly through sort of the process of drug development. But when you have a cancer like AML, like we're talking about, so we're focusing on NPM1, somatic mutation positive, and FLT3 positive as well. So in the US, there's on average, it fluctuates 19 to 21,000 on average each year diagnosed. And so if you take that small pie, 30%, and then within FLT3, it's 40%, you're talking about like 2,500 people in the US. So take such a small amount of people, and you're trying to, and these are huge decisions, right? Transplants over $100,000 to do a transplant. It's a big deal. It changes people's lives. And so you want good data. And so if you don't have big number of patients, you're going to want to use as much as possible uniformity in understanding the underlying biology. And that's where our assay really comes in play. And then also the issue with AML, unfortunately, it's often a terrible diagnosis where the survival is very poor, right, of 28%. So hopefully that will change with like all of us in this room. We can change that eventually, and we're making strides. But for now, I think the issue with what I hope to convey is we have to work together, and Invivoscribe really wants to help create basically a standard that we can come around and really hammer down signal ratio. So just to hammer home transplant in the US, there's only about 3,300 transplants for AML in the US, which wouldn't be that bad if it was spread across many just 1 or 2 transplant centers in the US. But if you look, in the US, there's only about 180 transplants centers that worked in AML. And of that, there was only a handful that were over doing 2 a week. So if you're like, trying to take these very sick patients, trying to base your decisions on maybe your own lab developed signal ratio, you may not have much clinical experience in trying to get the best outcomes for your patients. So I think when people think about have sometimes strong ideas about certain things, you really want to get into the weeds about what condition are we using, what's the assay's purpose. And with acute myeloid leukemia, it's a very small number of patients that are very sick. So you don't have much time, and we all sort of have to work together. And then lastly, in sort of the why is the therapeutics, right? We need to identify the people that have FLT3, and we need to do it quantitatively really well, but also qualitatively very well and quantitatively very well with a signal ratio. And there's basically some drugs that have already been FDA approved, and some are still on their way. And I think that's a testament to basically that these people are very sick, and there's concerted effort and a lot of money coming into it, and also understanding the underlying biology. So we know the ratified trial basically this showed that there was an overall survival hazard ratio of 0.76, and which led to their approval. And then Stellis recently just released their basically final findings of the phase 3 trial, the Stellis trial, basically showing the 33 or so prediction of relative risk reduction with that drug. So that's the why. If you can come out of this meeting thinking like, why LeukoStrat CDx, you want to think about the therapeutics, the prevalence of AML, and the importance of getting the signal ratio right. So next, let's talk about what we did. And this slide seems simplistic, but we can all appreciate in laboratory medicine, often simplistic. There's a lot of hard work that goes into these. And why I have this here is our guard band analysis. We spent a lot, like when I go into the lab and I talk to the people that really feel like this is their baby and they own it, they're really proud of the stability studies that we came into, making sure this assay was right, and the guard band analysis to make sure that you're not running this lab on the edge. Like there's a lot of good work that came in. And so they looked at different guard band analysis for different dilutions, the MMC layering, how you layer it down, where you're grabbing the sample from. They looked at the temperature of the centrifuge to make sure that that wasn't playing a role, and that it sometimes can vary, and that's not going to affect the analysis. We looked at the digestion, and basically we just changed the different variables. And for example, just one of the tests, the replicants for the layering, was more than what one publication did for their whole round one of their entire validation with like 9 labs. Like we ran hundreds of replicants for the layering, and that's what some studies are just based on. So it just talks about how this assay works, and there was a lot of work that went into it. Another thing about signal ratio, allelic ratio, that I didn't appreciate until like I talked to people that really are in the know. You'll hear, when you read a paper, you'll read about allelic ratio all the time. But it's actually the signal ratio that is being reported. It's the signal of the mutant over the wild type, the intensity of the signal. And why we can't say allelic ratio is to get allelic ratio, you have to really almost go to single cell, looking at the cytogenetics, pulling that out, and then doing layered sequencing on that. And that's just not done. And so we talk about signal ratio because it works. These are essentially equivalent if wild type and the amplicon of the internal tandem duplication are about the same, but that's often not the case with this condition. These, as you guys know, this internal tandem duplication can be quite large, and that's why this allelic ratio is not equal to the signal ratio. So next, that was a little bit about the nuts and bolts. So for that, if you can remember the guard band, we did a lot of work with stability studies and making sure that the assay works all the time and it's reproducible. The next is this. I have this slide up for the ratify because it took years and it took many countries and multiple oncologists to make sure it happened. And because AML is so rare, that's why I have it. It takes a lot of work to get, unfortunately, the deaths and to see the curves separate. Like to move on, like as you guys may know, this trial, there's a really interesting article in Blood Advances that Stone wrote, and he wrote about how these lines started plateauing, and they estimated to get enough deaths to meet their initial preliminary final like endpoints. It would have gone. The study would have gone to 2026, and you can appreciate a pharmaceutical company with their intellectual property does not, and the patients do not want to wait years and years to get this right. So why I bring this up is we often need to use this trial and to keep this uniform because things change as well. Like for example, this trial, they were essentially patients weren't being bone marrow transplanted, and then they started to, and they had to do censoring and uncensoring to sort of see that the basically the hazard ratio is 0.78 in an interim analysis instead of waiting for the final endpoints. Another thing too, I brought this slide up because again, like it got approved because of the 0.78 overall stratified. But gosh, this just always kind of like made me a little bit concerned is they haven't quite figured out the high and low on the FLT3 ITD. And the clinical trial assay is different from our CDx. And you wonder, like, was there a little bit in play with using high or low, or was it just the patients weren't high enough so it wasn't powered enough? But it's just, you would think that if you're going to spend that much money, that much time, you want to maybe use a universal assay from the beginning. This graph I showed basically, here's the publications, the Ratify trial, and then this is showing basically our clinical trial assay positive, clinical diagnostic LeukoStrat positive, and essentially you're getting the same survival. And this is in the non-censored for stem cell transplant. Okay, then now we're going to dive into the backbone of the lab validation that they used for the Ratify trial, the CalGB 10603. What they did is they did 8 rounds of clinical testing to quality control, where they sent basically 50 samples they prepared centrally, and they sent 25 samples were analyzed by each lab, looking to see how much agreement there was between the different labs that participated in the trial. And what they found was a little bit concerning. So what they found is that first of all, it's difficult to, when you get a clinical sample, there's no like gold positive, like you have to sort of arbitrarily assign what's actually right. And they define that as the median for each sample over all performing analyses. So they said that's the correct one. And then what they did is they looked at the FLT3 ITD high, which again is defined differently. In the Ratify trial, they use 0.7 versus other labs use 0.5. And unfortunately, the median error was 7.1%. It was higher when you get down to the LOD of 0.05. They said 0.06. It's 13.4. That improves if you run the assay in triplicate, which isn't done like usually clinically. So they had to run it 3 times to try to get something that worked. And so what they unfortunately did is, so remember back to how that the high low is not really working out quite right. Well, they found that 89 of 379 values were wrongly assigned to the above or below cutoff of the high or low allelic ratio, which is not, I don't think, acceptable. Like, this is like, extremely expensive, using multiple different clinical trial assays, and you wonder if they could have gotten the binning better with the LeukoStrat. Their conclusions was, and these are the authors, is this first report of round-robin testing for FLT3 mutation shows that using a standardized protocol, the qualitative assessment of FLT3 mutation is feasible with high accuracy. But that's just the qualitative. Like Real Abbott, they have the IDH one. Like a lot of these FDA approved use a qualitative yes/no binary, but I think what you'll see more and more is machine learning. It's going to really depend on the clone and the prevalence of that clone in the underlying biology. And they say, however, the assessment of the FLT3 allelic ratio clearly shows a considerable variability, which can be reduced by using a triplicate analysis, which isn't done clinically, but still has to be taken into account, especially in patients evaluated for stem cell transplant, which is $100,000, right? And for further standardization of FLT3 testing appears highly warranted. And I think that's LeukoStrat. So here's some own work we did comparing different clinical sites, which are in gray, and our site in IVS in red. And I just want to show you the distribution that we're fairly tight, except for one. I mean, we're not perfect, right? But the distribution, you can imagine if you're using multiple different labs, there's a bigger spread there than what could have happened if there was just one lab performing it. So I think more than anything else, it speaks to maybe if you're going to use a trial, use the same assay for the whole trial, particularly when you have such critical enrollment, which Jeff talked about, which many people struggle with in cancer, right, is enrollment. So this next is what we have published in the past, showing the Deming line, which is a statistical method using, essentially you can help compare one lab assay to another. The clinical trial assay, essentially, in our CDx, has basically a good regression, both in the ITD and the TKD. Next, you can read the technical sheet. I don't know if you are on the plane, you have nothing to do. It's a good read. Or the summary effectiveness data, if you're interested. It's also fun. It has a pretty picture. Goes good on a PowerPoint. But anyways, this just shows some of the work that goes into this FDA submission. This basically shows the strategy using 9 panel members, different lots, different machine instruments, and essentially seeing the reproducibility, the precision, right? Invivoscribe is all about improving lives with precision. Because it's easy to do it once. It's hard to do it worldwide, year after year, patient after patient, right? And so this just shows you like 27 replicates. This is, like 200 replicants. And the coefficient of variation, or the degree, the measurement of variability, was acceptable under 25%. It's quite low. And you're just not going to see these number of replicants. Basically, this is a lot study and also instrument study. The next one shows basically a between operator and between run, like are the people good as well? And again, you see, look at the volumes that you need for FDA submission, just go far surpass, like basically CLIA CAP guidelines on what is sort of acceptable benchmarks. Again, I think regulatory was really happy with these next 2 slides. They actually use clinical data. It's one thing, those were cell lines. This is clinical data showing, again, they diluted against a wild type. So it's clinical sample diluted across a wild type, showing medium positive, low positive, high negative. The coefficient of variation should have been less than 25%. The TKD data really shows minimal variability, as well as the ITD sizes. The variability, again, it was acceptable. To be fair, if you read the very fine print, at 217, the variation was high. We actually had to modify that. It, again, is well below the 25%. So to sum up, remember the why, which is basically the prevalence, the therapeutics, and the What was the third? You guys remember? Anyone? The single ratio. Good job. And then the box, A+2 business development. And then the next is think about guard band and all the hard work you have to do to make the assay work over time. And then finally, here's some real life data and how the assay works.